Hello and welcome to this video on grade 11 biology. This is Preetinder Kaur for Perfect Scores, and we are doing the different parts of a cell. In one of the earlier videos, we have already covered the first few parts of a cell. We did information on the plasma membrane, some organelles like the endoplasmic reticulum, the vacuoles, lysosomes, and the Golgi apparatus. And in this one, we are going to do the remaining parts of a cell, starting with the mitochondria first of all. And before we get started, don't forget to visit our website, that is perfect-scores.com, for more such videos in a more organized manner. And if you have any valuable suggestions or feedback, you can share them at perfectscores89@gmail.com. The mitochondria, the organelle that you can see, its number varies from cell to cell. They're also known as powerhouses of the cell because they store energy. Now, if the cell is performing lots of physiological activities, it will have more of these mitochondria. And if it is performing less of the activities, it does not need so many mitochondria. And as you can see, the shape is a little like a sausage or a cylinder with two different membranes, the inner membrane and the outer membrane. Now, the space between both of these membranes, this space that is not shown in the diagram, this is known as intermembrane space. So let me write that down, intermembrane space. And the central part is known as the matrix, and these extensions of the inner membrane that you can see, these fold-like structures, these are known as cristae. Now the cristae, they have an important function. They increase the surface area for the activities to take place. Now the two membranes, they have their own specific enzymes that are associated with the function that the entire mitochondria performs. Now mitochondria, they are sites of aerobic respiration. So that is where the respiration takes place. Aerobic means in the presence of air. And they also produce cellular energy in the form of ATP. And we'll be doing more about these processes in detail later on, what is ATP and how it is formed. And the reason why they are called powerhouses is because of this. They help in production of ATP, which is actually the cellular unit of energy. So that is how these are produced. The matrix, it also has a single circular DNA molecule. So about the matrix, it has a single circular DNA molecule. Additionally, it has a few RNA molecules, that is ribonucleic acid. And then it has ribosomes, the 70S unit. I hope you remember from the previous video where we discussed about the prokaryotic cell. We did ribosomes. They come in two units, 50S and 30S, and when they combine, they form a unit known as 70S. And lots of other components are also there that are needed for synthesis of proteins. So these other components are also present in the matrix. And how the mitochondria process each other, how do they divide? They divide by fission. So that is an important situation that you need to know of, that the division and the replication of mitochondria or formation of new mitochondria takes place through a simple fission process. So that is what you need to know about the mitochondria at a basic level. The next important cell organelle is known as a plastid, and here is an example of a plastid. This in front of you, you can see a chloroplast. The plastids are found in all plant cells and euglenoids, and it's very easy to observe these plastids under a microscope because of their huge size, means compared to other organelles. And they also have some specific pigments. For example, chloroplasts, they have a pigment called chlorophyll that gives the plastids a green color. So similarly, different colors are imparted. Now, based on the kinds of pigments, they can be identified and classified into three groups: chloroplast, chromoplast, and leucoplast. Now, the chloroplasts, they contain chlorophyll pigment and they are green in color. And it also has some carotenoid pigments. So these two kinds of pigments are present in chloroplast, and they help to trap the light energy that the leaves of the plant especially they are getting from the sun, and this light energy is useful for photosynthesis. In chromoplasts there are only carotenoid pigments, and these are all fat soluble. So fat soluble carotenoid pigments are there. I'll give you some examples. So these are two examples. One is carotene, one is xanthophyll, and this can give the plant lots of different colors like red, orange, or even yellow. The leucoplasts, these are colorless plastids. They do not have any color, and they have lots of various shapes and sizes, different kinds of nutrients. Now among the leucoplasts you will be able to learn about amyloplasts, which is a kind of leucoplasts. These have starch in them, for example in potato. Then one more kind of leucoplast is eleoplast, that store oil and fats. And then a third type is eleuroplast, which helps in storage of proteins. So that is about the leucoplast, chromoplast, and chloroplast. And now we'll go ahead and describe the chloroplasts in a little more detail. So now let's do a little bit more about the chloroplasts in detail. Now you need to know that the chloroplasts, they are usually found in the mesophyll layer of the leaves and the plants. That means the different organs of the plants. And in these mesophyll cells, maximum chloroplasts can be formed, and they are lens shaped as you can see in this diagram, or they can be oval or spheroid also. They have a variable length that can range from 5 to 10 to 2 to 4 unit, and the number it can vary from 1 or it can also be 20 to 40 in number per cell. For example, there's only one chloroplast in one cell of Chlamydomonas, while a simple green algae can have about 30 to 40 chloroplasts in one cell in the mesophyll as well. Now, just like the mitochondria, the chloroplasts also have two membranes. So one is an inner membrane, the one is an outer membrane, and then there is a space between them, that is the intermembrane space. Now, of the two membranes, the inner one is less permeable. So that needs to be mentioned here. That is less permeable. The space that is there inside the inner membrane, that is called the stroma. Means just like the matrix was there in mitochondria, the chloroplasts have the central space called the stroma. In this stroma, you can see a number of flattened sacs called thylakoids, stacked one above the other, and each stack or each group is known as a granum. Now these thylakoids, they are arranged like piles of a coin, and in addition there are some kinds of tubules called stroma lamellae that connect the thyloids of different grana. So this you can see this structure over here, the connection between different grana, it's done by the stroma lamellae. They connect the thylakoids of the different grana. The membrane of the thylakoids, it has a space known as lumen. So if you look at this being a thylakoid, inside this is the lumen. And then these thylakoids are arranged. This arrangement, this stack becomes known as the grana. Now it also contains small double-stranded circular DNA molecules. So let me write it down over here. Small double-stranded circular DNA. And apart from this, it also has carbohydrates. It has ribosomes. These are present. And it helps in synthesis of carbohydrates and proteins. So synthesis of carbohydrates and proteins. And chlorophyll pigments, they are present in these thylakoids. So these thylakoids, they contain the chlorophyll pigments. The ribosomes of the chloroplast, they are smaller. That means they are 70S. As compared to the cytoplasmic ribosomes that are 80S. So these are 70S ribosomes that are smaller than the 80S ribosomes made in the cytoplasm. So that was about the plastid, especially the chloroplast. And the next important thing that we have to do is the ribosome. Now ribosomes, if you remember, they were formed on the endoplasmic reticulum also. These are granular structures and they were first observed by a person known as George Palade. And that was in the year 1953. They're composed of ribonucleic acid, RNA, and proteins, and they do not have any membrane around them. So these are not the membrane-bound organelles. Now in eukaryotes, the ribosomes are 80S with the exception of the chloroplast. In the chloroplasts, the ribosomes are 70S. And in prokaryotes, the ribosomes are 70S. Now you might have wondered, what is this S that we are talking about? S here stands for Swedberg's unit. And it stands for the sediment coefficient. That means it's indirectly it's a measure of density. So both 70S and 80S ribosomes, they are composed of 2 units. I hope you remember that we talked about 70S being composed of 2 units, that is 50S and 30S. So similarly, the same happens for 80S also. They are also composed of 2 different units. So that was about the ribosomes. The next important cell organelle that we need to discuss is the cytoskeleton. And within the cytoskeleton, lots of things are there. Lots of filamentous proteinaceous structures are present. So filamentous and proteinaceous structures. That forms the cytoplasm, and that forms the cytoskeleton that is there in the cytoplasm to give its structure and support. And lots of functions are performed, but basically it is mechanical strength or mechanical support, strength, and maintaining the shape of the cell. Maintenance of the cell shape. So that is the main function of the cytoskeleton. The next important part is the cilia and the flagella. So the cilia and the flagella, they are basically hair-like outgrowths of the cell membrane. And they are small structures that work like oars of a boat. So remember, cilia are smaller and they help in movement of the cell or any fluid that is around the cell. Flagella, they are a little bit longer than the cilia, and they're also responsible for cell movement. Now the flagella is there even in the prokaryotic bacteria, but that flagella is totally different from the flagella that we are about to discuss, which is the flagella of a eukaryotic cell. Now the study of these two kinds of organelles, that is the cilia and the flagella, has been done through electron microscope, and it has been found that they are covered by a plasma membrane. And this goes for both of them, both the cilia and the flagella. The core, that means the central area, is known as axoneme. And they have lots of microtubules that are running parallel to the long axis. This usually has about 9 pairs of doublets. Of radially arranged microtubules in a pair of centrally located microtubules. So such an arrangement is known as 9 plus 2. So I'll draw a diagram so that it's more clear. So this here is the central tubule, the centromicrotubule, and these are the peripheral microtubules. So if you want to, let's say, label them, these will be the central microtubule, and these will be the peripheral microtubules. And remember, they are always in pairs. So you can see there are 9 of them here, 1, 2, 3, 4, 5, 6, 7, 8, and there should be one more here. So let me draw that. So here it is. And between the central microtubule and the peripheral tubule, there is also a sheath. So this is the sheath. And outside this, there is a plasma membrane. Okay, so let me label it. That is the plasma membrane. And then there are extensions coming out from these, which are known as radial spokes. And then there are small bridges that are there between these microtubules. These bridges that are here, these bridges are known as interdoublet bridge because they are present between two doublets. And there are lots of bonds as well, not just these bridges. So I would like to show them like these, like this. So that is the complete structure, quite a colorful one in this case. That's the internal structure of the cilia or the flagella. So now you can see that the axoneme is there, which is the central part. It usually has 9 pairs of doublets. They are radially arranged in a kind of a tubule, and that is known as the peripheral tubule. And then there is a pair of centrally located microtubules, and this structure is the 9 plus 2. The central tubules, as you can see, they're connected by bridges, and they're also enclosed by a central sheath, which is drawn by this green color over here. And this is connected to one of the tubules of each peripheral doublets by a radial spoke. So this spoke should be extending up till here, the central sheath basically. So there are 9 radial spokes, and the peripheral doublets, they're also interconnected by linkers known as interdoublet bridge. Now both the cilium and the flagellum, they emerge from centriole-like structures called the basal bodies, which we'll be covering next. So let's go to the next cell organelle, which is the centrosome and the centrioles. So centrosome is basically an organelle that contains 2 cylindrical structures known as centrioles. So when we draw diagrams, when we talk about cell division, we basically draw them as these two. These are the centrioles, and there are structure around them, and this entire thing becomes one centrosome. So they are surrounded by this amorphous pericentriolar material. So this is the amorphous pericentriolar materials. And both the centrioles, they are actually perpendicular to each other, which you cannot see in this two-dimensional diagram, but they are actually perpendicular to each other, just like a cartwheel. So if one is placed like this, the other one will be placed like this. They're actually perpendicular to each other. So what exactly are these made up of? Now they are made up of 9 evenly placed peripheral fibrils of tubulin protein. That is the name of the protein. 9 peripheral fibrils, and each fibril, that is a triplet. That means it has 3 fibers in it, and adjacent triplets are also linked. The central part, the proximal region of the centriole, is proteinaceous. So whatever I'm talking about here is about one of these. Let's say this one. So the central region, it's made up of protein, and it's called the hub. and it's connected with the tubules of these peripheral triplets by radial spokes. So the connection to the fibrils is by radial spokes that are also made of protein. So the centrioles are the basal body of the cilia or the flagella, and the spindle fibers that also help and give rise to the spindle apparatus, which is useful in cell division in the animal cells, which we'll be doing in later chapters. So right now that is all that you need to know about the centrosome and the centrioles. The next important structure that we need to do, the next important cell organelle, is the nucleus. So let's get started with this. Now, nucleus as a cell organelle, it was first described by Robert Brown. He was the first one who discussed the nucleus, and later the material of the nucleus, it was stained by the basic dyes. It was given the name of chromatin by Fleming. As you can see, the structure, it has a nucleolus, which is surrounded by a nucleoplasm, and then the nuclear envelope is there that has an outer membrane and an inner membrane, and then chromatin, which is the inner material in the nucleoplasm, it has heterochromatin, euchromatin, and then ribosomes are there on the surface of the nucleus, and nuclear pores are also there. Now the interphase nucleus, that means nucleus of a cell when it is not dividing, it has extended and elaborate nucleoprotein fibers known as chromatin. So let me just put it down. It has chromatin, which you can see in the form of fibers over here. It has a nuclear matrix known as nucleoplasm, and it has nucleoli. That means nucleolus, the nucleus of a nucleus. Now, electron microscopy has shown that the nuclear envelope also has two parallel membranes with a space of about 10 to 50 nanometers, and this space between them, that is known as perinuclear space. Perinuclear space, the space between the outer and the inner membranes of the nucleus, it actually forms a barrier between the materials inside the nucleus and materials outside the nucleus in the cytoplasm. Now, the outer membrane is usually continuous with the endoplasmic reticulum, and it also has ribosomes on it, and there are lots of pores also, which are basically formed by fusion of its two membranes. So that is what these pores are, nothing but fusion of the two membranes. Now these nuclear pores are actually the passages through which RNA and protein molecules, they move into and outside the nucleus. These are those junctions in both directions. Normally there's one nucleus in each cell. Sometimes there can be more than one nuclei in one cell. Some cells, for example, the blood cells of many mammals, they don't even have a nucleus. Even sieve tube cells of the vascular plants, even they do not have a nucleus. And then comes the nuclear matrix or the nucleoplasm that contains the nucleolus and the chromatin. Now nucleoli are the spherical structures. The content is continuous with the rest, and it does not have a membrane. This nucleolus over here, it does not have its own membrane. It is not membrane bound. And this is a site for the synthesis of active ribosomal RNA that is formed in the nucleolus. And larger and more numerous nuclei are also present in cells that carry out lots of protein synthesis. Now, from your 10th class biology, you might remember that in the interphase, that is when the cell is not dividing, the nucleus has lots of fibers of nucleoprotein. Those fibers are called chromatin. But during different stages, instead of chromatin, the nucleus, they actually show the chromosomes. Right, so let's do that. That is the next topic that we need to do. So here is a diagram of the chromosome that you can see. These are present in the different stages of the cell division. So let's study what chromatin actually contains. It contains the DNA and also some basic proteins that are known as histones. Now there are some non-histone proteins also, and there is a little bit of RNA also. So that is the composition of the chromosome or the chromatin as it is present in the interphase nucleus. Now one single human cell has about so much of DNA it could be stretched till 2 meters, and that is distributed among 46 chromosomes. So that is for humans. We have 23 pairs of chromosomes. And we will be studying the details of how DNA is packaged in the form of a chromosome, but that will be in grade 12, not in grade 11. Now every chromosome, it has a primary constriction known as a centromere that you can see, on the side of which you have the disc-shaped structures known as kinetochores. So these are the kinetochores, the centromere, and these are the kinetochores. Let me write it down again, kinetochores. Now based on the position of the centromere, chromosomes are of 4 types. So these are the 4 kinds of chromosomes that are present. The metacentric one has a middle centromere that has 2 equal arms of the chromosome. So let me label it as metacentric. The next one, the second one, it is known as submetacentric. And the third one, which is known as acrocentric, the centromere is situated close to one end, forming one extremely short and one very long arm. So that is acrocentric. And the last one that you can see, in which there is a terminal centromere, that is known as telocentric. So these are the 4 kinds of chromosomes that are there according to their shape. Now sometimes the chromosomes may have a kind of outgrowth known as a satellite. So they have a secondary constriction and then they have a satellite on it. So this becomes the satellite, and this stalk-like structure is known as a secondary constriction. Now as you can see, the centromere itself, it is a primary constriction over here. And these are the satellites, as you can see. So it's actually part of the arm itself, but it looks like a small fragment. So that was about the chromosomes, and the last part of the cell organelles is the microbodies. And these are nothing but membrane-bound minute vesicles. They contain lots of enzymes, and they're present in both animal and plant cells. So that is about the main cell organelles that we have discussed, and lots of organelles have been discussed in the previous video also, so don't forget to watch that one. And thank you so much for watching this video. Don't forget to visit our website, that is perfect-scores.com. You can like and share us at Facebook, watch more videos on YouTube, and if you have any suggestions, you can mail them at perfectscores89@gmail.com. So thank you so much for watching.